Deep in the Arctic tundra, scientists reintroduced mammoth genes into living elephant cells for the first time. It wasn't fiction or fantasy. It was a real experiment aimed at reviving extinct traits. Gene editing, once sci-fi, now guides nature's direction instead of simply observing its mysterious course. CRISPR technology lets researchers cut and paste DNA with surgical precision, like rewriting life's original manuscript. But as these synthetic edits expand, one haunting question emerges. Is this still nature or something else? In China, goats have been edited to grow more muscle and yield higher quality wool per animal. In the US, scientists engineered mosquitoes that can no longer carry malaria, changing ecosystems with every hatch. These aren't mutations born in wild storms. They're designed in labs by human hands, in silence. The Mexican axolotl, famous for regenerating limbs, now fuels gene research for regrowing human spinal cords. But when humans edit frogs to glow or pigs to grow organs, where does wildness truly remain? Nature once meant untouched wilderness, raw ecosystems guided by time, struggle, and blind adaptation. Now we breed coral that resists heat, bees that fight viruses, and salmon that mature twice as fast. None of these are hypothetical. They already live in waters, forests, and commercial labs around the globe. We've created cows that feel less pain and chickens with gene switches controlling their muscle-to-fat ratio. Biotech companies call this progress, but traditional ecologists see an eroding line between living systems and design. Is a bird still wild if its DNA was tailored to resist pollution or urban noise? What about wolves bred for docility to help rewild forests without threatening livestock and local farmers? These questions aren't future speculation. They're echoing across conservation boards, research ethics panels, and policy courts. In Florida, genetically modified mosquitoes are already battling invasive species with lab-altered fertility genes. In Australia, labs are building heat-proof kangaroo grasses to outlive the coming climate extremes. We are not just watching nature evolve anymore. We are directing how it changes, step by step. For endangered animals like the northern white rhino, lab-grown embryos are their last flicker of survival. Yet some argue that saving species synthetically might sever their deep evolutionary roots. Biological rescue becomes bioengineering, and wildness becomes a human-controlled variable, not a wild one. When synthetic DNA spreads through natural populations, wild ecosystems may never be fully wild again. We've already seen hybrid trout outcompete native species, not from evolution, but from hatchery gene manipulation. Nature's arms race once played out over millennia. Now it updates faster than your smartphone firmware. But what happens when engineered traits escape their test zones and mingle with unsuspecting wild populations? Could a drought-resistant gene in lab corn spread to invasive weeds and create unstoppable agricultural chaos? Or could it save entire food chains from collapse during future mega-droughts and soil degradation events? The duality is stunning. Human innovation may preserve nature or unravel it one small tweak at a time. Synthetic biology offers hope for biodiversity, but also risks of uniformity, imbalance, and ecological shockwaves. It's no longer a question of if we'll change nature, but how far we're willing to go. Some scientists dream of reversing extinction entirely using preserved DNA to bring back the thylacine or passenger pigeon. But reintroducing these species into today's ecosystems could trigger unknown ripple effects in food chains. Are we playing God with nature's canvas, or merely speeding up what evolution would do eventually? Even conservationists are split between letting species die naturally or modifying them to survive humanity's damage. Genetic rescue might protect elephants from ivory poachers by removing tusk genes entirely. But does a tuskless elephant still represent the same wild majesty as its ancestors once did? In a world where life is sculpted in labs, nature's future may lie in artificial resilience. And as forests burn, oceans acidify, and animals vanish, engineering may become the last tool we have left. But before we call it conservation, we must ask, are we saving nature or replacing it? And if everything is touched by human code, can it still be called wild at all?